Thank you very much, Jan. And hello, everybody. Um, so essential biodiversity variables and why I'm talking about this today, it's not a concept that's necessarily very broadly known yet. It's been around. The first publication on essential biodiversity variables came out in 2013. And yet it is in some ways one of the central parts of our activities in SAEF, and so that's why I'm going to introduce it to you today and talk to you a little bit about it. So if we think about SAEF's title, Securing Antarctica's Environmental Future, and the title of the proposal had the byline, an informatics approach. I mean, why is that the case? And the point is that well, at least I see, I guess everybody who was involved in the proposal might see it a little differently, but I see that informatics approach as informatics being a vehicle for bringing about a step change in the quantity and quality of Antarctic biodiversity data, and the relevance of those data for policy and management. So central to our question, our science in SAFE, at least particularly in theme 2, is how is biodiversity generated and maintained? And we're taking a long-term and broad spatial scale type of approach to that question. But of particular pertinence, of course, is how that biodiversity is responding to current change and being able to predict how it is likely to respond to near future and future change, with the purpose of intervening where we can, managing that biodiversity where we can, or at least planning for the future of the region and our activities in it. But the informatics component of it brings in the additional questions here, which are sort of what information on biodiversity do we need across the Antarctic region in order to manage it effectively, or to formulate policy to manage it effectively? And how do we best go about getting this information? And this is where the concept and initiative that is essential biodiversity variables comes into it. So if we think about monitoring biodiversity and what we understand about it, monitoring biodiversity, of course, has a very long history. It developed a little bit of a bad reputation in the 80s, and there was quite a lot of disinvestment in monitoring in the 80s going into the 90s as a consequence of it being perceived as a scientific activity of that was using up a lot of resources, but not actually delivering on policy and management relevant questions. But we now know differently. We now know how essential it is to have information on biodiversity in order to understand how it's changing and the consequences of that change for human well-being. And so we need to look at how ready are the data that we have for addressing the sorts of questions that I outlined in the previous slide. So this is a collation of information across different sources of biodiversity data. So this is in the little colored dots here. So we get biodiversity data from inventories such as citizen science-based surveys, for example, like the US breeding bird survey. We get biodiversity data from expert input such as expert maps, monographs. We have inventories which are plot-based, such as vegetation surveys, and we have inventories, national inventories, for example, marine fish surveys. And so we have a lot of biodiversity out there, and it's coming from a range of different sources. And if we look at the value of those data for reporting on biodiversity and change in biodiversity, we can think of it, if we look at the top left of the figure here, as being of low value or of high value. And we can assess each of these different data sources according to a range of criteria. So, how many records do we have? What are the spatial grain of those records, the temporal grain of those records? What is the taxonomic scope and coverage? Are the data shared? Are the metadata shared? When the data is collected and stored, is there a quality control mechanism? Do the data adhere to agreed data standards, for example? And then we can assess where these various sources of biodiversity lie according to these criteria for generating high quality data. And without going into the detail, you can very quickly see that we can draw basically three conclusions from this. One, data are coming from a range of quite different sources. Two, each of these data sources have different properties, and against some of these criteria, they actually perform quite well, but against other criteria, they don't. And so the essential conclusion here is in biodiversity monitoring globally and looking at the adequacy of biodiversity data globally. Generally speaking, biodiversity data are not ready for use in monitoring and for informing policy. And yet there is a lot of biodiversity data out there. So the problem we sit with is this diverse set of data, each of which has various strengths and weaknesses. And ideally, we want to be able to capitalize on this wealth of information, but we're currently not in a position to do so. Another aspect that comes into play, of course, is that even when we have the data, data suffer from a range of different biases, some of which are inherent to the taxa involved, and some of which are more consequence of the way in which the data are collected. So if we look at how confident we can be in terms of the inferences we make based on modeling biodiversity data, if we just look at the top left here, so we want to be able to infer change from biodiversity data, and our ability to do so is very dependent on the grain at which those data are collected and collated. So when the data are available at a very coarse grain, our ability to infer change is very low compared to when it's available at a fine grain. If we look on the right here, not only do we want to be able to infer how biodiversity is responding to various drivers, we want to be able to generalize our findings across taxa or across regions, but our ability to generalize inferences is of course also dependent on how the taxonomic coverage of the data we have, how representative it is of the data that are out there. And then of course, our ability to accurately infer and or predict is dependent on where in the world you're looking at. So there are a lot of data biases. In Papua New Guinea, for example, our prediction uncertainty is quite high compared to prediction uncertainty for biodiversity in the UK. Both a consequence of the amount of biodiversity, but also a consequence of the properties of the biodiversity data and the extent of biodiversity data available across these two areas, for example. And then there's also taxonomic differences. So our knowledge of global bird distributions far exceeds our knowledge of global plant distributions, for example. So clearly, globally, there are well understood caveats that undermine our ability to predict the responses of biodiversity to global change and the consequences of those responses for us and the environment. And it's worth thinking, I think, in an Antarctic context of where our Antarctic biodiversity data sit with regards to these various properties, biodiversity data properties. So it's not only data quality and data quantity that's important, of course. We can have vast quantities of data and yet still not effectively inform policy and management in a relevant way. So we need to put in place monitoring that is policy and management relevant. And this is a social process where we look at the explicit knowledge in the sense here that comes from biodiversity data and biodiversity science. We need to think about communicating that knowledge. and also engaging with stakeholders and managers, of course, so that they can help to formulate and identify the priorities. And via those priorities, we as a group of scientists then look at what biodiversity data are a priority and how do we model those biodiversity data in order to address the needs of these stakeholders. So particularly importantly, up the top here, engaging with managers, governments, policymakers, and so on, is critically important to create that so-called authorizing environment. So if policymakers buy into the process from the beginning, they're more likely to take heed of the outcome of any sort of biodiversity assessment or biodiversity monitoring outcome. So essential biodiversity variables, where do they come from? Well, they actually come from, emerged from the idea and the success of essential climate variables. Some of you are probably aware of these, but there is a global climate observing system called GCOS, and an essential climate variable is defined as a physical, chemical, or biological variable or a group of linked variables that critically contribute to the characterization of Earth's climate. And it was interesting to see how these essential climate variables came about. It was in about 1988. There was the IPCC came about, and it was through the activities of the IPCC, partly driven by UNEP, the United Nations Environmental Programme, and the World Meteorological Organization, that quickly realized that climate is monitored in different ways around the world. There's different focus on different variables. And if we were going to be able to report on the global state of the climate and climate change, then it was absolutely essential to harmonize monitoring across the globe. And so that's where this idea of essential climate variables came about. And so the whole rationale was to ensure that the observations that we're making, measurements that we're taking, and the information that is needed to address climate change issues are obtained and made available to all potential users. And it was also born of the recognition of the strong need for sustained observations of these essential climate variables. So there's clearly a need to invest in long-term data generation, analysis, modeling, and delivery for policy and management purposes. So the essential biodiversity variables were modeled on the same rationale, or came about with a similar rationale, and were modeled on the same sort of process. So you may know about Geobond, the group on Earth observations, a biodiversity observation network. And Geobond's mandate is essentially to work towards improving the quality, quantity, and value of biodiversity information globally. And Geobond has three core focus areas. One is the development and implementation of essential biodiversity variables. The other is supporting the development of biodiversity observation networks from national scales to regional scales and globally, all of which with a very firm eye on producing policy relevant outputs. So Geobond has a mandate through the Convention on Biological Diversity, and it works very closely with IPBES. If we look on the right here, the Intergovernmental Science Policy Platform on Biodiversity and Ecosystem Services. There are enough acronyms here to drown in. But the CBD is essentially this global scale policy. IPBES runs scientific assessments. Geobond is responsible for data for facilitating, enabling, and promoting data integration, analysis, and delivery. And this is where essential biodiversity variables play a key role. So what is an essential biodiversity variable? They're defined as the derived measurements needed to study, report, and manage biodiversity change, focusing on status and trends in elements of biodiversity. So one of the factors that has plagued the biodiversity monitoring for a long time is a sort of conundrum that it feels like everything needs to be monitored. And we know that it's practically impossible to monitor everything. So how do you make this decision about what to monitor? And this is where EBVs come in. So just to give you an idea, if we take a particular problem here, and let's say our concern is the conservation of whale populations and whale species, and we're generating data, and there's some threats on whale populations. These could be ship strikes, could be tourists disturbing breeding grounds, and so on, could even be whaling. And we have data coming in from various sources. We have satellite data, which gives us some information on whale distributions. We have actual presence occurrence record observations, and these together, for example, can be integrated to generate information on whale distributions. And via a particular suite of models, we can come up with global distributions of whale species and their populations. And we might integrate those actual species observation data with data such as ocean color, which tells us about chlorophyll, which might inform where we would expect whales to feed, for example. And obviously sea surface temperatures might also enable us to predict future movements and or current movements of some of these marine species. So we have our problem sketched out here. We have the data sources on the outer rim, but those are the raw data. Those raw data need to be collated in a form that is able to actually provide you with distribution information that is sensible. And then we would use those sorts of collated raw data in a range of interpolative and predictive models to produce policy and management relevant information on whale distributions. So there are 6 essential biodiversity classes and 21 sort of proposed essential biodiversity variables underneath that. So the classes are essential biodiversity variables on genetic composition, species population, species traits, community composition, ecosystem function, and ecosystem structure. And I'll talk a little bit about the species population EBV class, partly because this is the class of EBVs that are probably best developed at the moment conceptually and also to some extent in terms of implementation. So just EBVs have a definition, and you can look at the species distribution. EBV can be defined as the probability of occurrence over contiguous spatial and temporal units for the global extent of a species group, which consists of one to many members. That just recognizes the fact that we might be interested only in the distribution of a single species rather than a whole suite of species. And similarly, the species abundance essential biodiversity variable, I won't go into that because I see we are running out of time. But another feature of essential biodiversity variables is you can think of it as a many-to-one and a one-to-many type of relationship. So the many-to-ones, we have essential biodiversity variables in the middle here, and the many-to-one comes in at, it reflects the fact that we have these multiple data sources, all of which need integrating, and that integration requires an extent of automation, modeling, data quality assessment, and then modeling to produce EBVs. So this type of modeling is modeling to interpolate or gap fill, is modeling to reduce data biases that are well known, as we talked about earlier, and it's also modeling to forecast or predict how that biodiversity is going to respond to change. So we have multiple inputs into single EBVs, for example, the EBV of species distributions. And the important thing is that these are not static data layers, but the idea is that we can update those distributions over time so that we're working towards spatio-temporally dynamic information that then feeds into a range of policy and management needs. For example, producing biodiversity change indicators, but any other sort of policy or management reporting need. So this is why a single EBV, such as species distribution, particularly if you look at the definition of that EBV as encompassing multiple species over space and time, can then be used to feed into multiple different policy needs. So that's why it's a one-to-many, going from EBVs to their application. So the vision, I guess, for essential biodiversity variables is that they encourage this integrated knowledge generation, analysis, and reporting cycle. And that together, these 6 EBV classes, supported by thematic bonds such as biodiversity observation networks that focus specifically, for example, on alien and invasive species like Grant talked about, but also national and regional biodiversity observation networks support the data input into these essential biodiversity variables, and the scientific community then assists in producing policy relevant data products. So essential biodiversity variables are really a community endeavor. There's a broad community of agencies and researchers involved in doing this. Geobond, again, a lovely acronym soup here, GBIF, CBD, IPBES, TADWIG, which is the data standards group for biodiversity data, map of life, but importantly also country nodes and biodiversity observation networks and scientists. Now, in case you think this is really pie in the sky, there's been massive progress made over the last couple of years, and I'm just showing some of the papers from the areas that I'm more closely involved in when it comes to essential biodiversity variables, which are the species populations variables, and also particularly applying this to biological invasions. And so some of the modeling that I was talking about now would be some of the models that Grant was talking about in his talk, for example. And so, the work that's been done includes things like improving data standards for alien species in this case, looking at how we can ensure that the EBV approach enables monitoring across spatial scales. We've produced a data register called the Global Register of Introduced and Invasive Species, which is a living, breathing, active tool, which was not necessarily inspired by EBVs, but where the whole production line of EBVs has actually facilitated this very policy relevant product. We've also done some work, for example, looking at how existing data infrastructures such as GBIF and the Atlas of Living Australia can contribute to the EBV data generation process. We've produced national guides, for example. And as I said, this is just a small section of the work that's been done to develop the concept of essential biodiversity variables and start to implement them and use them in policy reporting. So now we need to think about what does this mean in an Antarctic context, and how do we draw on the capacity and thinking and developments that are happening under the banner of Geobond and the broad range of scientists involved, and through the EBV concept, and how do we use this in Antarctica again to really bring about a step change in the quality, quantity, and value of biodiversity in the Antarctic via SAEF. Thanks.